Okay, everyone, hello. Today we're going to be talking about the concept of microbial ecology. Okay, tell a story about the ecology of microbes of Earth. So, if you'll allow me to be a little corny for a moment, let me tell you a story before we do this. A long time ago, in a galaxy far, far away, or this galaxy right here, we had a series of planets, a solar system, circling around a single star. Okay, and on one of these planets, it was a hellscape. It was a fiery ball of magma for quite some time in these early years. As a mass had developed here, rock had formed a large concretion as more meteors, asteroids, junk from outer space collided with this growing object, and it grew in mass. And as it did, it retained a lot of heat over the years. Okay? And this planet was far from habitable for quite some time. For millions and millions of years, this planet had an incredibly inhospitable landscape. It was fiery hot. It had a complicated and deadly atmosphere. Okay, and it was generally not conducive to life for a long, long time. But eventually, life did emerge here as the planet cooled slowly over millions and millions, really billions of years. And this planet, it went through a period of transformation. It was natural as major changes happened to this planet and its geology, and also its atmosphere. It developed, and eventually this planet, pretty miserable as it was, became conducive to life in a certain form. And so here, somewhere on this planet, where it's a bit unclear at the moment, but somewhere here, life did arise. And from this place, life grew, proliferated, and developed eventually to the point where it encompassed the world collectively. It became widespread. Okay? And these early pioneer forms of life, they did not live the same way that we are most familiar with organisms living. They survived mainly on the energy that they got from inorganic resources. Things like gases in the atmosphere that they could make use of, minerals in the ground or dissolved in the water, compounds that were free floating in the water or being ejected from, at the time, still a very violent Earth. And along the way, these forms of life devised some interesting strategies for making use of those unusual resources. For one, these early organisms figured out synthesis pathways that allowed them to put together organic compounds, oftentimes from inorganic resources. And this was really essential for them to be able to grow themselves and reproduce, to make more copies of themselves. And other organisms, all organisms, developed respiration pathways as well, which was used to convert energy sources that in some cases were inorganic, in other cases were organic in nature, and convert them into something that was more useful, more widely accessible, or more easy to store. And so these two forms of pathways are very important for early life. One particularly interesting group of the synthesizers that figured out some really interesting strategies were the photosynthesizers, right? The organisms that could do photosynthesis. Of course, we're familiar with this as a concept. This is a method of converting water and carbon dioxide into sugars and also oxygen as a byproduct, using sunlight as a source of energy to make that reaction happen. And this was really handy because you could take the sugars that came from this reaction, you could rip them apart and do more things with them. You can use them as an energy source, but you could also use them to build important structures like lipids, which you could use to make more of the phospholipid bilayer, and expand the cell and grow it, prepare for reproduction. So there's a lot you could do, and that's just touching the very, very surface of what you can do with these sugars. But so these organisms were figuring out new tactics, right? Photosynthesis, as I mentioned, produced a really interesting toxic byproduct, and that was oxygen. But that was all right? Not a big deal. Because deadly oxygen, this compound was something that was scarce in the environment at the time. It made up a very, very small percentage of the atmosphere. And so it was not something these organisms would encounter very often. And so these organisms could produce this waste and let it go free into the environment without really having to worry too much about it coming back to them and being a problem. It would just diffuse into the environment and go away, never to be heard from again. And for a time, that was kind of how this stuff worked. It was a very harsh existence, but it was a prosperous one for the organisms who lived here. That this life on this planet proliferated. It evolved. It diversified into all of these different habitats across the world and adapted to the varying conditions that were present here. And the planet, in turn, went through its own changes in response to these evolving organisms and the processes that they did over the course of their lives. So we have this really interesting thing that is developing now at this time where the carbon dioxide is being used quickly and frequently by these organisms that are doing photosynthesis. And so the concentrations are decreasing. And at the same time, oxygen is increasing in abundance as this waste product is produced more and more. And as it did so, this change, the reduction of carbon dioxide and the increase of oxygen content in the atmosphere, it cooled the Earth a little bit faster than it normally would be, right? It made Earth more comfortable, at least by our standards today. And eventually, it got to the point where water wasn't just existing in a liquid medium, but it could also exist as ice in some parts of the planet. However, the cumulative effect of these organisms was piling up. Oxygen, as it built up in the atmosphere, was becoming a problem now, many, many millions of years after the ones who created the strategy started to produce it. And so this toxic compound built up in the atmosphere, it was a building problem, one of the organism's own making, and eventually it reached a tipping point, a point where now it was too late, and now the organisms were facing the toxic effects of this oxygen buildup. After a certain point, oxygen's built up to a concentration that was high enough to kill many different sensitive life forms. Lots of organisms that were particularly sensitive to the oxygen concentration, they died out completely. And so there was a mass extinction event that followed here. That very few places on the Earth actually remain relatively safe from this devastating condition. And this is a phenomenon that became known as the Great Oxidation Event. It was so prolific in evolutionary history for this planet that it still bears a scar of a sort on this planet and in the evolutionary record of the species that live here. But it may seem like that was probably the end of it, but it was not. Ultimately, the survivors of that cataclysm survived, and they adapted and evolved to live in those new conditions. The planet was now different. It was rich in oxygen, and now they had to deal with it. They had to live in that environment and ideally make use of it if you could, right? It was now a very abundant resource, whether they liked it or not. And so these organisms developed new strategies to utilize oxygen, right? Here we have the aerobes, right? The aerobes that developed this strategy. On the other hand, we had some organisms that kept the old ways, as you might think of it, right? The aerobic way of life. That they avoided developing these new strategies and instead changed their behavior. They instead went and survived in areas that were naturally low in oxygen. They stayed in areas where they could be safe from these toxic byproducts that were now so abundant in the environment. Other organisms invested in those strategies. These organisms avoided them, right? They invested only in the things that they already had, okay? And so we have this splitting that happens where some organisms stayed anaerobes and many more abandoned that lifestyle to live amongst the oxygen and utilize it, becoming aerobic, oxygen-breathing organisms. So, obviously, I'm talking about Earth, right? There's no secret there. This is, what I described here, is the leading theory on early Earth development, okay? That we have this really interesting history of microbial life that establishes early in Earth's geological record, although still almost 1 billion years or more, multiple billions of years late from its formation. And so these organisms develop, they arise somehow. How they arise, we aren't entirely sure. But as they do, they live their lives, and by them simply living their lives, they shape the earth and change it because of the things that they do, right? The products that they make, the resources that they use. And in doing so, the earth changes under their influence. And so we have these microorganisms to thank or blame for creating the cool, habitable earth that is rife in oxygen that we live in today. That's really helpful, of course, for us, because that's all that we know. So this great oxidation event is something that occurred long before plants even existed. This far predates plants, and it's attributed mainly to cyanobacteria, photosynthesizing bacteria species, right? Of which many of them still exist today. An important element here that I think can be gleaned from this story is that no organism is an island. The lives of the organisms that occupy this planet impact other things besides them, whether we talk about direct or indirect influence. That you have organisms that may interact directly. Maybe you have organisms that steal food from one another, like a lion stealing a recently hunted gazelle from a pack of hyenas, right? That kind of thing can happen in the Serengeti, and that's a very direct interaction. But you also have interesting conditions like what I described of microbes changing their environment through their lives, and it turns around and impacts other organisms besides themselves. That the toxins that they create through their survival poison other organisms that cannot utilize them. Or, in the case of oxygen, provide an important resource today. That some organisms that are producing oxygen, such as the cyanobacteria, they provide those essential resources that we today benefit from. The Earth is a semi-closed system when you think about it. And of course, things are not really leaving Earth. Things can arrive on Earth. Sometimes we get, comets that pass by, and particulate from those comets can decorate the Earth, can land on us. Of course, meteors and meteorites that officially land on Earth. We have things that can reach Earth, but things don't usually leave Earth. So things that are trapped within the gravitational field of Earth are here. And there's only so much matter that is here. There's a finite array of resources that are here that organisms have to utilize. And as organisms live and they utilize those resources in turn change in a few interesting ways. They change form. They change location. They change how accessible they are to different organisms and who can access them. And that very much has to do with a number of interesting biological processes that the lives of organisms that utilize them have. This ultimately, we're talking about something that's ecology. We're talking about ecology here. The relationship between organisms and the impacts that they have on their environment and each other. Okay? That's ultimately what ecology is. It's the study of relationships between organisms and their environment. And that is very much what happened in early Earth, that we use that example. It's very much something we see today in many different forms, including in a microbial world. And so let's talk a little bit about these things, right? So we have life that falls into two main groups based on their ecological roles. We have autotrophs and heterotrophs. Autotrophs are living organisms that create organic molecules from inorganic carbon sources. Okay? So think about these like the photosynthesizers, that they take inorganic resources like carbon dioxide, right? Carbon dioxide is an inorganic molecule. Even though it contains carbon, it is an inorganic molecule. And sunlight and water, right? One is just an energy source, and the other one is another inorganic molecule, right? Water. And they utilize an enzymatic process to bind these things together, to react them with the sunlight that excites them and create sugar, right? Glucose, an organic molecule that now they can use to do other things. Heterotrophs, on the other hand, can't do this, right? They can't do that. They have to acquire their resources from organic sources, right? So heterotrophs, they're not building these things from scratch. They are not making organic molecules from inorganic sources. Instead, they have to get these resources from organic sources, which typically means consumption of a living or dead organism, right? Autotrophs can further be broken up a little bit into different categories. Phototrophs are the photosynthesizers. These are the organisms that utilize sunlight to do this. But we also have some really interesting chemotrophs, right? That they are chemistry-assisted autotrophs. So they utilize a more unusual inorganic compounds. Like, for example, hydrogen sulfide. There are organisms that live in the deep ocean vents in the furthest reaches of our planet, in the deep ocean. And they live right on the cusp of these volcanic vents that are erupting hydrogen sulfide gas, among other things. It's extremely hot. It's extremely... it's also weirdly cold. It's a weird dual kind of environment where a mere inch in any direction can have hundreds of degrees of temperature difference. And they survive by feeding off of the hydrogen sulfide that's kicked out of these vents. So those are chemotrophs, special bacteria that live down there. And so another common categorization that you'll often see is producers and consumers, right? They are in many ways synonymous with autotrophs and heterotrophs, respectively, right? Producers make resources from scratch, from organic sources. Consumers must consume resources that are made by producers to survive, right? They will either consume producers or other consumers, right? Or in some cases, dead material from either, right? So I want you to make sure that you know both of these terms, producers and consumers, and also autotrophs and heterotrophs, okay? A lot of heterotrophs, because of their lifestyle, they end up being predators or grazers or parasites, right? Predators are organisms that actively hunt, capture, and consume live organisms. They kill them and then consume them, or consume them while they're alive. Grazers are consuming mainly live organisms or immobile consumers, right? Mainly live autotrophs or immobile heterotrophs. So think about this like a giraffe is eating leaves of a tree, right? And also, the act of doing this will not necessarily kill the plant. It will cause the plant injury for sure, but it won't. It's not a guaranteed thing that it will definitely kill them, right? So it's just a general difference in the way that we think about consumers and the way that they eat. Parasites, of course, are organisms that feed on other organisms without killing them, right? But we can also have variety here, right? There are some organisms that act as parasites that will kill their hosts, right? Or cause, in some cases, significant injury or symptoms to their host, in which case we call them pathogens, right? And then some heterotrophs will eat mainly dead things or loose organic matter, right? Things that have dissolved. Maybe we're talking about waste material. Maybe we're talking about particulate from an organism like shed skin, or when an organism dies and is torn apart by a predator, the bits and pieces that remain, that these loose organic materials can become food to decomposers and detritivores, right? Where decomposers and detritivores, they feed mainly on detritus or rotting matter, right? That's what really a detritivore is, okay? Decomposer will usually eat mostly a dead, recently dead, or not so recently dead organism, right? So producers and consumers, they all ultimately, even though they have different strategies for achieving them, they have the same needs, right? They all need the same things. Those basic building blocks of life, the biological molecules, those being carbohydrates, lipids, amino acids, and nucleotides. These are the things that build every cell at its most base level, okay? And so all living things, whether they're autotrophs or heterotrophs, they're going to need these things. It just differentiates how they get those things. Do they make them themselves, or do they get them from someone else? These molecules, they are organic molecules. Okay, I mentioned this before. These are carbon-based compounds with abundant carbon atoms and also with abundant CH bonds. Okay, that they have a lot of carbons attached to hydrogens and carbons attached to carbons. Okay, that's what we call an organic molecule. And so it should be obvious then why carbon dioxide, even though it has a carbon molecule in it, is not an organic molecule, right? There are no hydrogens attached to it. All of life, ultimately, is in a constant pursuit of these carbon-rich molecules. And so as they pursue these things, as they create them, as they use them, as they excrete them as waste or lose them, the carbon within them exhibits a cycle that moves through these living things and also through the environment. And this occurs as the compounds break or they bind into new compounds, new molecules. The bonds between carbon molecules and carbon atoms and other things break and allow them to change shape, form into something new, right? In some of these forms, the carbon is accessible to different forms of life. But in other configurations, it's not. It is unavailable, inaccessible to them. And so some organisms will have to take these things and change them in some way, or they will have to change as a reaction to some chemical in the environment in order to make them available again. So let's talk about that, the cycle of carbon, right? What does that look like? Well, carbon is going to move through our environment in a few different ways, right? It starts, though, in the atmosphere and with the autotrophs. So as I mentioned before, we have photosynthesis as this tactic that some organisms have devised in order to take carbon from the atmosphere as carbon dioxide and bind it together into glucose. In this way, we have this transformation. We take 6 carbon dioxides, 6 water molecules, and we, with light and enzymes, right, and chlorophyll, right, we smash them together and we form glucose and oxygen as a result, right? The organism especially cares about the glucose. Some organisms care about the oxygen, some don't, all right? And so this is a transformation. We have the alteration of form in these carbon molecules, carbon dioxide molecules. They lose their oxygen. They're stripped of some of it, and it binds to these hydrogens from the water, right, and some oxygen, and together they form glucose, right, a molecule that is suddenly accessible and usable to certain organisms, effectively every organism. Every organism is capable of using glucose. Okay. At the same time, autotrophs are trying to devise, they have to utilize energy, right? What they're doing, in part, in capturing carbon dioxide as CO2, part of that is them creating molecules they can use to build up their body with, right? Sugar has multiple uses. You can string them together to form long carbohydrates. You can break them apart and form lipids from them. You can use them as a base to make lots of different things, right? But another really important feature that we're all very familiar with is that sugar contains energy. And these organisms can break that molecule apart and use the energy stored inside it to create ATP, which is a more accessible energy molecule, something that can be more widely utilized across the cell. Okay, that process that I just described, the taking of glucose and converting the energy inside, I should say, into ATP, that is called cellular respiration. Cellular respiration, right? Of course, there are aerobic and anaerobic varieties that I talked about before, and they are similar in some key ways, mainly that they move the energy trapped inside of glucose into ATP, which they can then take the ATP and use it elsewhere. And they will both produce carbon dioxide as a byproduct, right? As a waste product. And so autotrophs and heterotrophs alike, they are doing respiration. They all will do that process. It's just that autotrophs will make the fuel for respiration from scratch. Heterotrophs have to acquire that fuel elsewhere, right? They have to eat autotrophs or other heterotrophs in order to get that fuel to power that process. So we have this cycle, right? We can take carbon dioxide, make it into sugar. We can take sugar, we can break it apart as well and make it into carbon dioxide. But we can do a third thing that I alluded to earlier as well. We can take that carbon and we can store it, short-term or long-term, as other molecules. We can turn it into all of those 4 biological molecules. Other carbohydrates, of which glucose is one of them, lipids, amino acids, and nucleotides, right? A creative cell can make all of these things, right? They're used ultimately to build the cell, to develop it, to have it grow, to remain part of that cell, hopefully continuously, ideally until death, right? It can also be used to make new cells as the cell reproduces. It may even be used to make compounds that the cell will intentionally release, like toxins, or in the case of, say, the glycocalyx, we have a gelatinous slime, right? That is meant to be outside of the cell. So it will eventually kind of wear away. It's not unexpected. And so in some particularly long-lived organisms, like these massive sequoia trees here, the carbon can remain trapped for years and years, centuries even, maybe even millennia in some cases, even after the organism dies. And so we have what becomes the carbon cycle here, okay? The carbon cycle that is mainly split between photosynthesis and respiration. So carbon dioxide through photosynthesis turns into glucose, which can become other materials. They are taken inside of these organisms and utilized, or sometimes released into the environment, sometimes as organic carbon. Other organisms will eat that and take those compounds up into them and utilize them as they see fit. And both these organisms will be doing respiration to release that material as carbon dioxide again, right, into the atmosphere. We can also have situations where materials can be trapped and it can decay, right? As it decays, it can off gas, it can change forms into different things, or it can remain fully trapped, right? Become locked in place and stay buried for millennia and millennia. Maybe it will even transform into a fossil fuel, right? Fossil fuels were once living organisms that died, were buried, and through a variety of chemical processes that lasted millions and millions and millions of years, they changed form so much that they became these long chain carbons that contain a lot of energy when they're burned, right? They burn and release a lot of energy. And so, and by the way, when we burn them, guess what we're releasing? Carbon dioxide, right? So we have this cycle that develops, this being the carbon cycle, okay? And currently, we are feeling the impact of changes to the carbon cycle. The carbon dioxide output is far greater today than our carbon dioxide fixation, right? The storage of that material as solid organic matter, right? As glucose, as lipids, as proteins, and so on, right? And this today is due primarily to significant burning of fossil fuels. And also, at the same time, an aggressive destruction of existing carbon stores, like logging forests. That's really not helping the situation. These cycles, like I said, they are cycles, which means that they are cyclical, right? But not necessarily in an equal way. It can be cyclical but slanted, where more of one kind of reaction is happening than another kind of reaction. And that will carry with it changes that develop over time, over years, over centuries, millennia, and so on. This concept that I'm describing, the movement of these nutrients around like this is the basis of something called nutrient cycling, which occurs in all of these biologically important nutrients. We see this in one form or another, okay? And so in this way, we have a cyclical movement of matter that occurs between organisms and their environment, different parts of the environment, say aquatic environment and terrestrial, or terrestrial and atmospheric. It can move between multiple organisms when one thing eats another, for example. And it can also change states. It can change molecular composition. These transformations, these movements, this is what defines nutrient cycling. And we will find that there's a carbon cycle, as we said, there's a sulfur cycle, there's an oxygen cycle, there's a nitrogen cycle, there's all these different kinds of cycles that any of these compounds that are utilized that have biological applications will often find a complex cycle in which it is involved. At some of these points, these nutrients are going to be unavailable to certain or all organisms. And in other situations, the nutrient will be actually usable by them. Not by all of them oftentimes, but by some of them, right? So there's a lot of really good examples of this, but I think we're really going to talk about the nitrogen cycle, okay? We've already talked about the carbon cycle, we'll talk about the nitrogen cycle. Because it is a rather complex one and it's very, very interesting. So the nitrogen cycle is similarly complex to the carbon cycle. We mentioned last lecture that nitrogen was very abundant in the atmosphere as nitrogen gas, as dinitrogen, right? That these compounds, though, this molecule, it's not really all that accessible. Not really abundant as an organic molecule, okay? Nitrogen as a gas is very, very unusable to most forms of life because those bonds are really difficult to break. Nitrogen actually has 3 covalent bonds that hold it to its other atom. And so it's pretty tough. It's a hard bond to break. Otherwise the material remains kind of locked away, unable to be accessed by most forms of life. But some bacteria have cracked the code on this and have figured out a way to transform N2 gas into other compounds that are actually usable by them and by other organisms. That process is called nitrogen fixation. Specifically, nitrogen fixation is the conversion of N2, right, nitrogen gas, into NH3 or ammonia, which is a much more accessible form for this. Okay. So this is a process that, it is a metabolic process, so it has a number of steps to do this. And it also takes quite a lot of energy. It takes 16 ATP for an organism to take one molecule of nitrogen and make it fixed, quote, fixed into NH3, into ammonia. Okay? And it's mediated by an enzyme called nitrogenase. Remember, all these enzymes, their names are most usually an indicator to their function, right? Nitrogenase. Nitrogenase is an enzyme that works with nitrogen, right? So these organisms, we call them diazotrophs. Okay, these bacteria that are nitrogen fixers. Right, nitrogen fixers. You'll see nitrogen fixers as a more common name, I would argue, than diazotrophs, though, in just the general media. Many of these organisms are free living, right? Such as the nostoc species that exist over here. This is a form of cyanobacteria that is a pretty well studied organism, all things considered. Nostoc is also very common. Okay, we find it pretty widespread. And these organisms are free living. They don't really associate with other organisms directly, but they are a photosynthetic organism, a photosynthetic cyanobacterium that will live just kind of as sludge sometimes on a surface and be actively doing photosynthesis and also producing fixed nitrogen, right? And some of these organisms, some of them though, have really interesting mutualistic associations where they very closely partner with some other organism. And there's an exchange of resources. So a great example of this is the nitrogen fixation that occurs in legumes. Okay, so if you've ever eaten peas and beans and what else? A lot of different plants fall into this, right? But particularly beans and peas. They are members of the legume group. And legumes are unified by an interesting association that they have with bacteria. We call them rhizobia. Rhizobia, this group of nitrogen-fixing bacteria that will grow in these little nodules that you see lining the root structures of these plants. And so these nodules, they're packed full of bacteria that are actively doing nitrogen fixation. And they do this, they live inside the roots because it's a trade of sorts. The plant is going to provide the bacteria with the products of photosynthesis. which they're not doing. The very important resource of organic carbon, accessible carbon. And in exchange, the bacteria are going to trade fixed nitrogen. They're going to trade ammonia for these products. So the plant desperately needs a constant supply of nitrogen. The bacteria needs a constant supply of carbon, especially in sugar form. And because they have what each other needs, they can exchange that, and they both benefit from it, right? So it's a really interesting partnership. Here's another demonstration, some diagrams of what that looks like, right? It's really interesting. They actually penetrate the cells themselves, and the structures of those rhizobia. They form these nodules that are kind of variable. I believe it varies depending on species as well, right? So sometimes they'll look like little nodules. Sometimes they'll look more branching, like big inflated roots. As ammonia, as NH3, nitrogen-fixing bacteria can utilize the nitrogen, right? They can actually take that and they can do things with it. They can incorporate it into a lot of their essential biological molecules. The ones that really need that nitrogen are the amino acids and nucleotides. Both of these groups use a lot of nitrogens, right? A lot of nitrogen in these compounds. And these are really important because, of course, amino acids are the base of your proteins. Nucleotides are the base of your nucleic acids, which are your DNA, your RNA, your ribosomes, right? So very important for base function. Okay. These bacteria, at the same time as they're doing this, they also open the door for nitrogen utilization for other organisms. Because other organisms can eat the bacteria, in which case they steal its nitrogenous molecules. And the organisms can also access the ammonium itself, right? The ammonia itself, and use that to build other molecules. Sometimes that material is just simply let go and released into the environment at times. Sometimes a bacterium dies and is not eaten, and the stuff gets seeped out into the soil or the ground or the groundwater, right? And so it can become a resource that is utilizable. But at the same time, some organisms can't actually use ammonia. And instead, ammonia has to be converted further into something else in order to be used by these organisms. So that is something where nitrification comes in. Okay, this is where we take ammonia and we oxidize it. We introduce it to oxygen and produce either nitrites or nitrates. Okay, NO2 nitrite, NO3 nitrate. And so this is a job that is mostly done by bacteria. Okay, there are some other ways this can happen, but it's really a bacterial job. Okay, and these, we call them the ammonia oxidizers. These are the bacteria and archaea that perform this job. And some of them will only convert to NO2. Okay, they'll only take it to nitrites. And again, only some organisms can utilize that. For some of these organisms, that's all they need. And so that's all they're going to do with it. But some organisms will take that further and bring that all the way to a nitrate, which is more broadly useful, especially to plants. Nitrates to plants. And so once nitrogen has actually been absorbed and incorporated into an organism, it's going to regularly change forms. Okay. First we have the consumption of that material, and it's going to be digested and broken down into those nitrogen-rich molecules. And then from there, the organism is going to figure out what to do with those things. It's going to build them into other materials. Okay, the things that an organism builds are not necessarily made and then forgotten about either. Sometimes the organism will be recycling its own material. So it might take the amino acids, sorry about that, the amino acids of the protein and break them apart again to use them somewhere else, right? And then also when the organism dies, right? Organisms that break down the materials and the waste products of that organism are going to change forms, turn it into something else again, right? It's going to be digested and then reconstructed. And bacteria and fungi are very important for this process, right? The active consumption of organisms that have a bunch of these molecules in them, and also the breakdown, the decomposition of those molecules, right? And specifically the breakdown of these nitrogen-rich molecules into ammonia, right? Sort of returning them to an earlier state. So that is a process called ammonification. Ammonification. The breakdown of nitrogen-rich molecules into ammonia, right? Into ammonia. So the sources for this, typically proteins, DNA, RNA, loose excreted waste materials, and so on, right? So we are essentially going backwards in this cycle. And then we have denitrifiers, okay? Denitrifiers. A common path for a lot of this nitrogen is fixation, then nitrification, and then consumption by something that then takes it and processes it into something more useful. Sometimes that stuff is then excreted as waste, which might even be ammonia before it gets processed or ammonified, right? But there are other ways that nitrogen could just return to being atmospheric nitrogen, N2. And so some bacteria specialize in this tactic exactly, where they can take nitrates, nitrites, things like that, and they can break them down. They can strip the oxygen from them, and they can turn them into just atmospheric nitrogen, okay, which is then released back out into the atmosphere. So these are the denitrifiers. These are the denitrifiers, organisms that are denitrifying the nitrates and nitrites, okay. And interestingly, this is a job that can be done by other nitrifiers. So some nitrifiers will also be denitrifiers. They will work the process one way and then work the process the other way, getting something new out of it, right? It's very interesting. So this process is really interesting. It's pretty multifaceted. You have a lot of different directions that nitrogen can move, okay? It can be easily integrated into these different organisms that you can follow it pretty aggressively along the path where you can say, okay, from atmospheric nitrogen, I understand how it can be taken up by rhizobia and turned into ammonia, and then something like a rabbit can eat that plant and turn it into something more useful to the rabbit. Maybe it turns into amino acids in a protein. Then that rabbit dies, and it is decomposed by bacteria and fungi that break down the proteins into ammonia or ammonium, which is just an ionized version of ammonia, and then that compound can be nitrified, right, into nitrites, maybe all the way into nitrates by other types of bacteria, and maybe it can be reassimilated into plants, or denitrifiers can release it as atmospheric nitrogen, in a sense, restarting that process. Okay? So these cycles, they're very important, and they occur widespread across the world and through, in part, the actions of organisms. There is an abiotic element to this. For example, lightning strikes can be an effective way to take atmospheric nitrogen and fix it, okay? But that's not something that happens very often. It's not something that produces tons of this material, and so it's not particularly leading the nitrogen fixation process by any means. Instead, bacteria are doing that, right? That is very much a bacterial job. So really, our ecology right? The interactions that we and other organisms have with one another and the environment are driving these processes. Even we are part of this process. We're animals too. We breathe too, right? As we are doing respiration, we are releasing carbon dioxide into the atmosphere that can then be used by another organism, like a nostoc cyanobacterium or one of these beautiful trees in this forest to create sugars and continue this process. Okay. If we're smart, we can actually utilize the natural processes that organisms do just by living their lives, and we can utilize those to improve our lives, right? This is the process of bioremediation in a nutshell, okay? The process of utilizing the ecology of organisms, particularly microorganisms, to clean or process environmental pollutants. Usually we're focusing on bacteria, fungi, algae, some plants, right, to do this process. And usually we're doing this as an effort to remove things that we have released ourselves, right? Pollutants that we have released into the environment either accidentally or intentionally. And sometimes we're using them to clean up unavoidable waste products, right? Things like the material that we produce, right? We poop, right? The fact that we are going to be making our own waste products, and we have to put our waste somewhere where it's not going to hurt us, and ideally not hurt other things. And we can utilize biological actors, right? Microorganisms to break down a lot of these processes, a lot of these waste products and pollutants that we create ourselves, even just by our living, right? And we can essentially clean those materials and prevent them from causing problems elsewhere, right? Which is really cool. This is particularly useful for us when mechanical or chemical methods to clean or prevent a spill or pollution fail, right? And so today we actually utilize many methods of bioremediation to do just this. So some of the compounds that we produce, again, either intentionally or unintentionally, they stay in the environment for a long time, right? Pretty long time. And some of these, when they're absorbed by organisms, can become what we call xenobiotics, right? These are pollutants that are foreign to an organism's system and can actually be kind of detrimental to their health at times, sometimes more severely than others. And in many cases, we don't even really understand the impact that these compounds will have on the organisms yet, right? A great example of that is DDT. Right? DDT was an insecticide that we sprayed kind of everywhere, particularly in North America. It was used elsewhere as well, but we sprayed it all over the place, because it was a very effective insecticide, and it was really good, especially as we were fighting malaria in America, in the southern reaches of the country, and other mosquito-borne illnesses. So we were killing all these insects and also a lot of crop pests and spraying the stuff everywhere, all over the place. But we didn't really realize that it stuck around for a long time. And it got taken up by organisms that were then eaten by other organisms who were eaten by other organisms. And it would make their way all the way through the food chain, ultimately ending up in animals like predatory birds, where these compounds would show marked health effects. In these birds, the eggs that they would lay, when they were so filled with DDT, they were fragile, immensely so. They were soft. And so when the bird would sit on their own eggs in order to warm them and incubate them, they would break them under their own weight. And by the way, a bird does not weigh much. These birds, even large eagles and things like that, they are not especially heavy animals. And so, unfortunately, it did not take much for them to break these eggs. And so a very sharp decline happened in their population, and we created that problem. We were scrambling to deal with it, right? We sprayed this stuff everywhere, for quite some time, right? So now, of course, we changed our behavior. We don't use this stuff anymore, but this was very much a xenobiotic to a lot of animals, particularly those birds. And it nearly caused some of these species to go extinct completely, like the California condor and even the bald eagle, right? A very symbol of our country. It's just very interesting that was almost rendered extinct because of our own actions, right? Today, we are worried about a new xenobiotic, that being microplastics. And we truly do not fully understand the extent to which microplastics affect us. This is something that we are still learning about. And it's kind of scary because we are finding more and more that even our bodies are rapidly accumulating microplastics. And so, this is a case of xenobiotics that are affecting us today. So when we're talking about bioremediation, think about this. Consider an oil spill, right? A disaster situation, right? Oil is mostly organic hydrocarbons, right? It's what we talked about previously. It's carbons that are strung together and they're fairly high energy. They burn really well, but they're also hard for an organism to digest them. Most living things can't eat them or live off their energy in any way, right? They're just there. It is an inaccessible resource for them. But there are actually some bacteria that have devised a way to digest hydrocarbons. Great example of this is a species called Microbial hydrocarbonoclasticus. It's a huge mouthful of a name, but the name, right, refers to what it does, right? Marinobacter, it is an organism that lives mainly in marine environments. Hydrocarbon, talking about oil, and clastic means to break down, right? So this is a free-living bacterium that can be found in many marine environments, and it's known to digest petroleum products. So this species, right, is very widespread in marine habitats, but it is rare when it occurs here. And so it's probably not very active when an oil spill happens in a marine environment. The chemicals that are used to actually disperse the oil spills, we're finding out, are actually very inhibitory to their growth. So oftentimes when there's an oil spill, in order to prevent the oil from clumping together, because of course oil does not want to interact and mix with water, we will go and spray a bunch of compounds that will force the oil to dilute itself and then spread widely throughout the ocean, right? It's a compound. It's a method that we use to sort of like alleviate the problem, but it doesn't really address the biggest issue. And so this compound, a lot of these compounds that we use, these clearing agents, right? They are inhibitory to these bacteria. And because the bacterium is naturally quite rare, right, occupying only maybe less than 1% of the total diversity or the abundance of bacteria, in any given bit of water, right, it will take time for these organisms to feel to like make their presence known, right? Remember that there is a lag time. If we think back about the pattern of microbial growth, there's a lag time that starts before you have exponential growth to a point where the resources are sort of exhausted, right? Or that they reach a sort of cap to which they can survive in this environment. That takes some time to establish. And so, as an oil spill continues to be a problem, these bacteria, even if they're there, they can take some time in order to ramp up to the point where they are especially productive and really giving a true assistance to the cleanup method, right? So today, researchers are exploring how best to utilize a lot of these microbes, including this species, right? How best to like more effectively and efficiently clean up the oil spills when they happen, and methods to encourage that rapid growth of the bacterium. We also utilize bioremediation in our wastewater treatment cleanup, right? As we sort of deal with the material that we produce ourselves, right? Our own waste products. We are utilizing various strategies, including a cocktail of microbes that break down a variety of different materials in waste. So, it was really interesting because some of these jobs, they're really only possible by some microbes. It's not especially efficient or effective for us to try to do these ourselves with mechanical or chemical methods. The bacteria have already figured out a very effective method, right? So ultimately what we're doing is we're separating out solids and liquids and processing them separately with different rounds of digestion and breakdown by these microbes. And there's a few different kinds. There's two different kinds, really, of bioremediation. Okay, it falls into two categories. We have biostimulation and we have bioaugmentation. Biostimulation is when we're using nutrients to ramp up and encourage the growth or activity, metabolic activity of an organism that is doing a bioremediation job, right? Something that is already there, okay? So in the case of the oil spill cleanup example, those microbes are already there. A biostimulation strategy might be going out there during an oil spill and adding fertilizers that enhance and increase the rate of bacterial growth. We encourage the growth of the bacteria that are already there, and so we're essentially eliminating the restrictions that otherwise constrain the growth of those bacteria, right? Bioaugmentation is different. This is when we're actually adding microorganisms to a place that doesn't normally have them to perform this job. So in the case of our bioreactors and wastewater treatment plants, we are adding what is otherwise absent microbial community, right? That we add an assortment of specific chosen bacteria, archaea, things like that, to do these different jobs in these wastewater treatment plants. And so we were sort of adding interesting and useful nutrient cycling roles that were not being filled by some other organism at that time. And so, bioremediation, this is a really cool field. It's rapidly growing. It's a very rapidly advancing science. And we're sort of constantly developing new solutions to problems as we gain new problems that now need solutions. So, very important field that needs a lot of attention, for sure. And thankfully, it's starting to get that attention. Okay, everyone. That's all for today. I hope that you learned a lot, took a lot away from this. This is kind of a complicated chapter and an important one, for sure. So, anyway, please tune in next time for our next lecture. All right, take care.